Hi everyone, hello from Singapore. Thank you for taking the time to join us for today's event of Deep Tech in Asia Stem Cell Innovations, organized by SGInnovate. My name is Jodie from SGInnovate, and for those of you who may be more new to us, SGInnovate is a Singapore government-owned organization that invests in and helps build deep tech startups, talent, and community. We have our mission to help build deep tech innovations from Singapore and for the world. The World Economic Forum in 2020 opined that stem cells could be the medical innovation of the century. Stem cells, in their ability to self-renew and differentiate, could also have the potential for a myriad of medical treatments. Today's event focuses on presentations by leading stem cell scientists in Asia who have developed novel stem cell technologies for better patient outcomes. Our program today will start with a presentation by Professor Keiichi Fukuda, CEO of Heartseed Inc., followed by a short Q&A session before we start our second presentation and Q&A session with Professor William Huang, Medical Director of National Cancer Centre of Singapore. Without further ado, I'll now like to pass the time over to our moderator for today, Dr. Natasha Ng, President of Biotech Connection Singapore, for her to introduce herself as well as our esteemed panelists. Natasha, please.
Okay, thanks, Jodie. Good afternoon, everyone. I'm Natasha, and it's my pleasure to moderate today's event. So as Jodie mentioned, I'm President of Biotech Connection Singapore, and we're a non-profit organization that promotes life science entrepreneurship and provide a platform for innovators and stakeholders in the biotech ecosystem to come together. Besides that, I'm actually also a research fellow at A-Star in the Stem Cells and Diabetes Lab, and so today's topic is actually of great interest to me personally as well. So I would like to welcome our two guest speakers for today's event, Professor Keiichi Fukuda from Japan and Professor William Huang right here in Singapore. So let me introduce them first. So Prof. Fukuda is a pioneer of the cardiac regeneration field and has been at its cutting edge for the past 20 years. And he has made several contributions to both the fundamental biology of stem cells as well as how to regenerate cardiomyocytes and transplant them into the heart in vivo. He became chief professor of the Department of Cardiology in Keio University and has published hundreds of top-ranked papers as well as worked as an editorial board member for more than 10 international journals. He established biotech company Heartseed in 2015 and became CEO of the company. He is now planning to embark on a clinical trial for transplantation of human iPS-derived regenerated cardiomyocytes to patients with congestive heart failure. And this program is supposed to start this year in 2021. Right, so next I will introduce Prof. William Huang. So Prof. Huang is Medical Director of the National Cancer Centre of Singapore, Chair of the Oncology Academic Clinical Program of Duke NUS Medical School, Head of the SingHealth Duke NUS Cell Therapy Centre, and Senior Consultant of the Department of Hematology, Singapore General Hospital. Prof. Huang is also a faculty member of the Cancer and Stem Cell Biology Program in Duke NUS, as well as Clinical Lecturer of the Yonglulin Medical School. He has published over 100 clinical and scientific papers in the field of hematology and oncology. And without a doubt, he is a key opinion leader in this area, as well as the rising cell therapy field in Singapore. So we're very honored to have both Prof. Huang and Prof. Fukuda to speak to us today. So as Jodie mentioned, we'll start off with a 20-minute presentation by Prof. Fukuda, followed by a 10-minute Q&A with Prof. Fukuda before we move on to Prof. Huang. So if the audience, please share all your questions in the Q&A box, and I will try my best to address them during the Q&A session. Thank you very much. And I would like to hand over to Prof. Fukuda right now.
Good afternoon, ladies and gentlemen. So I will share the slide. So let's begin. Thank you, Mr. Chairman, for the kind introduction. I am Keiichi Fukuda, Professor of the Department of Cardiology in Keio University School of Medicine. I am also a founder and CEO of Heartseed. I'd like to thank all the organizing committee to give me a chance to present data at this fantastic meeting. I had been working in the field of cardiac regeneration for more than 25 years. Today I'd like to talk about clinical application of human iPS cell-derived purified ventricular cardiomyocyte transplantation for the treatment of severe congestive heart failure. A loss of cardiomyocyte often causes heart failure in various heart diseases such as myocardial infarction, myocarditis, sarcoidosis, and DCM. But cardiomyocytes cannot regenerate by themselves. So, in order to rescue the heart failure, we needed to transplant the cardiomyocyte. So we transplanted iPS cell-derived regenerated ventricular cardiomyocyte into the failing heart. If we can achieve this, we can regenerate the failing heart. In order to achieve the heart regeneration for the treatment of severe heart failure, we need to overcome multiple steps. This slide shows a multiple step for regeneration of the heart. Number 1, efficient generation of safe and high-quality iPS cells. Number 2, massive expansion of iPS cells. Number 3, selective induction of ventricular cardiomyocyte. Number 4, augmentation of cell division in developing cardiomyocyte. Number 5, purification of cardiomyocyte to eliminate residual iPS cells or non-myocyte. Number 6, development of the efficient cell transplantation method. Number 7, preclinical animal experiments to confirm safety and efficacy. This slide shows selective generation of ventricular-specific cardiomyocyte. Myosin light chain 2A, MLC2A, is a marker of atrial cardiomyocyte. Myosin light chain 2B is a marker of ventricular-specific cardiomyocyte. Most of the lab can generate cardiomyocyte, but this is a mixture of atrial and ventricular-specific cardiomyocyte. Heart-seed can separately induce either atrial cardiomyocyte or ventricular cardiomyocyte. As shown in this slide, these cells are also all green positive cells. Of course, as you can see, some of the cells are shown in red, but these red cells also stained with green. This indicated these cells are fetal ventricular cardiomyocyte. This slide shows transplantation of non-purified cells causes teratoma formation. ES cells and iPS cells are one of the most attractive cell sources in regenerative therapies for heart failure patients. However, if non-purified ES or iPS cell-derived cells are transplanted, they will form teratoma. Red indicated cardiomyocyte, and this slide shows transplantation of non-purified cells causes teratoma formation. ES cells and iPS cells are one of the most attractive cell sources in regenerative therapies for heart failure patients. However, if non-purified ES or iPS cell-derived cells are transplanted, they will form teratoma. Red indicated cardiomyocyte, and sky blue indicated undifferentiated iPS cells, and other colors indicated non-cardiomyocyte. If we transplant the cluster of these cells, they will form a teratoma like this. So, elimination of residual stem cells and purification of cardiomyocytes are necessary. This slide indicates the ideal strategy for cardiomyocyte purification. Red indicated cardiomyocyte, and sky blue indicated undifferentiated iPS cells, and other colors indicated non-cardiomyocyte. If we can make a medium in which undifferentiated iPS cells cannot survive and non-cardiomyocyte cannot survive, we can easily purify the cardiomyocyte. In order to achieve this, we had performed various types of experiments, and we can purify the cardiomyocyte. This slide indicated iPS cell-derived cells before and after purification of cardiomyocyte. The left-hand panel indicated two-dimension cultured iPS cell-derived cardiomyocyte before purification. And the right-hand panel indicated the cardiomyocyte after purification. These non-beating cells were non-cardiomyocyte and residual iPS cells. After purification, only cardiomyocyte could survive and continued spontaneous beating. The reason why residual iPS cells and non-cardiomyocyte disappear is like this. They strongly required glucose and glutamine as an energy source. So we depleted two major energy sources. Also, glucose and glutamine are important for cardiomyocyte, but if we depleted glucose and glutamine and supplemented lactate into the cell culture medium, cardiomyocyte can survive because cardiomyocyte can efficiently use lactate as an energy source. This slide indicated large-scale production of human regenerated cardiomyocyte by robot system. The right-hand video indicated the robot system to generate cardiomyocyte by robot system. We do not need a technician to do the cell culture. Using this machine, we can generate large amount of cardiomyocyte. The next step is cell transplantation. When we transplanted cardiomyocyte directly after trypsin digestion, cardiomyocyte can survive in the host heart. However, the number of survived cardiomyocytes are few. The survived transplanted cardiomyocyte by floating single-cell injection is less than 3%. Most of the cardiomyocytes were lost by backflow due to bleeding. We investigated how to increase the cell survival after transplantation. And we collaborated with some chemical company to make a specially designed cell culture dishes. This is a specially designed cell culture dishes. As you can see, this plate have a lot of small holes at the bottom. And using this specially designed cell culture dishes, we can re-aggregate the cells and made cardiomyocyte balls. This is cardiomyocyte balls. As you can see, each balls contain 1,000 purified cardiomyocyte. And the cardiomyocyte organoid can facilitate the cell survival after transplantation. This is a result of cell transplantation. In this case, we used mouse ES cell-derived purified cardiomyocyte balls into mouse heart. The left-hand panel indicated 3 weeks after transplantation, and the center panel indicated 8 weeks after transplantation. As you can see, the adult cardiomyocyte had beautiful striation. However, very interestingly, transplanted regenerated small cardiomyocyte have also striation, and cluster of cardiomyocyte can survive in the mouse heart. And very interestingly, as you can see, the size of the cardiomyocyte shows bigger and bigger after transplantation. Right-hand panel indicated immunostaining. Green indicated cardiomyocyte nucleus, and red indicated actinin, indicating cytosol, cardiomyocyte cytosol. The size of the transplanted cardiomyocytes shows physiological hypertrophy after transplantation. Lower panel indicated human ESL-derived cardiomyocyte spares into immunodeficient mice heart. Human cardiomyocytes were stained by red dye. As you can see, a human cardiomyocyte can survive in the mouse heart. Regenerative cardiomyocytes were transfected with luciferase cDNA. When luciferin is injected into the peritoneum, the survived luciferase-positive cardiomyocytes were detected from outside of the body. The upper panel indicated the single floating cell transplantation. The amount of luciferase-positive cardiomyocytes were very small. The lower panel indicated cardiosphere transplantation. The large amount of cardiomyocytes could be detected. The right-hand panel indicated a quantitative analysis. So this experiment strongly suggested that cardiomyocyte spheroids revealed high cell survival after transplantation. This slide indicated the immunostaining of the human iPS cell-derived cardiomyocytes transplanted into mice heart stained with anti-actinin antibody. The left and right-hand panels indicated 2 weeks and 4 months after cell transplantation respectively. The upper and lower panel indicated low and high magnification respectively. The transplanted human iPS cell-derived cardiomyocyte shows physiological hypertrophy and sarcomere structure were clearly demonstrated at 4 months. This slide shows the result of cell transplantation of the human iPS cell-derived cardiomyocyte into mice. Left-hand panel indicated 2 weeks after transplantation, and center panel indicated 4 months after transplantation. This is a co-immunostaining for myosin light chain 2A, green, and myosin light chain 2B, red. Myosin light chain 2A is an atrial cardiomyocyte marker, and myosin light chain 2B is a ventricular cardiomyocyte marker. As you can see in the panel, at 2 weeks after transplantation, some of the green cells can be observed. However, at 4 months, there are no green or yellow cells, indicating the transplanted cardiomyocyte fully differentiated into adult ventricular phenotype. The left-hand panel indicated the immunohistochemistry with anti-human alpha-actinin of the human iPS cell-derived cardiomyocyte transplanted nog mice. The transplanted human cardiomyocyte shows physiological hypertrophy and adult phenotype. Beautiful striation can be observed. No scar formation was found in the transplanted area. The right-hand panel indicated the video showing the beating heart tissue was observed at 1 year after transplantation of human iPS-derived cardiomyocyte into nog mice skin. Please take a look at neovascular formation supply the blood flow of these beating tissues. So human heart tissue is generated in the mouse skin. These findings indicated transplanted human cardiomyocyte into nog mice generated functional myocardium. Next, we had developed an original cell transplantation device. The tip of the needle does not have a knife. They were closed, but they have several side holes. Smooth tip of the needle does not damage the vasculature to reduce bleeding. and cells were transplanted as shown in the center lower panel. The video shows the overview of the transplantation. Cardiosphere floating liquid were mixed with ink. Please take a look at the injected site. There are no bleeding after transplantation. In this experiment, we had generated a cynomolgus monkey MI model and observed the effect of human iPS cell-derived cardiomyocyte transplantation on cardiac function and arrhythmia. In this case, we had generated myocardial infarction by ischemia reperfusion model at 14 days before cell transplantation. And we transplant 20 million human iPS cell-derived cardiomyocyte into left ventricular wall. As you can see in the left panel, brown area indicated the transplanted human cardiomyocyte. The right upper panel indicated changes in fractional shortening after iPS cell-derived cardiomyocyte transplantation. Red indicated human iPS cell-derived cardiomyocyte, and blue indicated control experiment. As you can see in this slide, transplantation of human iPS cell-derived cardiomyocyte improved cardiac function. The lower panel indicated the duration of ventricular tachycardia by Holter monitoring. In this experiment, we used 5 cynomolgus monkey model, and we had observed arrhythmia after transplantation. As you can see in this slide, only one animal revealed 9-minute sustained ventricular tachycardia at day 14 after transplantation. But the other 4 animals do not reveal significant arrhythmia. So these findings strongly indicated that human iPS cell-derived cardiomyocyte transplantation can improve cardiac function, but do not cause significant arrhythmia after cell transplantation. This slide shows the conclusion of the preclinical experiment. 1. We can selectively induce ventricular-specific cardiomyocyte. 2. We can purify cardiomyocyte with more than 99% purity, and residual iPS cell is less than 0.001%. 3. We had developed transplantation device with minimum invasion and bleeding. 4. The transplanted human iPS cell-derived cardiomyocyte formed mature ventricular phenotype, beautiful striation, and a new vessel formation. 5. The transplanted human iPS cell-derived cardiomyocyte survived for a long time without making any teratoma or other tumor formation in dog mice heart and skin. 6. The transplanted human iPS cell derived cardiomyocyte into MI-generated cynomolgus monkey heart improved cardiac function. The 9-minute sustained VT was observed in 1 among 5 monkeys. This is a protocol. First, in human clinical study, we will use HLA haplotype Homo iPS cells, which was generated by Shinya Yamanaka's institution, Kyoto Saira. This is the most frequent HLA in Japanese population. HLA 6 class match can reach approximately 17% of the Japanese whole population. The human iPS cell-derived cardiomyocyte will be transplanted into HLA-matched patient. This slide shows our future clinical plan. For first-in-human clinical study, we will directly transplant HLA-matched human iPS-derived cardiomyocyte into the heart muscle in patients with dilated cardiomyopathy in late 2021. For clinical trial, we will directly transplant HLA-matched human iPS-derived cardiomyocyte into the heart in patients with HFrEF due to coronary heart disease simultaneously with CABG in late 2021. Our global business partner is Novo Nordisk. Novo Nordisk is ranked 7th in the world in terms of market cap, well known as the best diabetes care provider. Net sales is 20 billion US dollars and 45,000 employees and 169 countries with affiliates. Their next major strategic focus is serious chronic disease, with such as heart failure and cardiovascular indications. Let's switch gears to our collaboration with Novo Nordisk. Here is the structure of our deal. We've licensed allogeneic iPS cell-derived cardiomyocyte or spheroids, regardless of indications, route of administration. Our clinical development starts in Japan, and we hope earlier approval in Japan utilizing accelerated approval scheme. As for commercialization, since Japan is our home country, Heartseed retained the commercialization right. We'd like to be a full-fledged pharmaceutical company in Japan, while we have to establish the necessary organization. We believe this structure is the best for us because we can utilize existing infrastructure of Novo Nordisk Japan, while we can pursue upside together. This slide shows a comparison with other global deals. Blue, red, and orange column indicated Staten and Novo Nordisk, Sigurion and Lilly, and Symbio and Dainippon Sumitomo, respectively. These are the very big deals. However, in case of Heartseed, a groundbreaking iPS-related deals with up to 598 million US dollars in upfront and milestone payment. Thank you very much for your attention.
Hey, thanks, Prof. Fukuda, for the presentation. And it's hugely exciting to see how far your technology has come, and the preclinical work you have presented is impressive. We definitely look forward to seeing the outcomes from your upcoming clinical trials. So great, we have time for some questions, and I see that there are a couple of questions on the Q&A box. So the first question, Prof. Fukuda, from Andre. What do you think is the size of the market for stem cell therapy globally, and in particular for heart treatment?
It's very difficult to say. We expect the very big market size because the number of the heart failure patient in Japan is 1 million. And the heart failure patient in the United States is 5 million. And all over the world, the heart failure patient is approximately estimated as 26 million. So the market size will be very huge. But it is difficult to say.
Okay, thanks, Prof. Fukuda. There is another question that there are a few more specific questions to the that's related to the technology. So one from Shifeng, what percentage of the transplanted cardiomyocytes leave the heart? Is there a chance that they implant elsewhere in the body?
Yeah. In our experiment, we had observed at least 50% of the cardiomyocyte can survive in the heart, and mostly 70% or like that. There is a big difference between floating cell, single cell transplantation and cardiosphere transplantation.
Okay, thank you. Another question that's related to the details of the technology. So how does the individual cardiomyocyte spheroids electrically connect to each other, as well as connectivity to the host, I presume after transplantation?
Yeah, we have already confirmed that the transplanted cardiomyocyte beats synchronously with the surrounding cardiomyocyte. We transfected G-camp into the regenerated cardiomyocyte and transplanted them in several different sites. But all the transplanted cardiosphere beat synchronously, and we can observe from outside of the heart. So we confirms electrical coupling.
Great. There are more questions coming in, but there's another one from Lynn. How was VT monitored and determined in your monkey experiment?
Yeah, in our hand, we transplanted human cardiomyocyte in various type of animals. When we transplant them into the mouse and rat, there are no VT. When we transplanted them into the pig, there is a strong immunorejection. At that time, we observed some VT. But when we transplanted the human cardiomyocyte into the monkey model, the number of arrhythmia is not so big, because we can selectively induce ventricular-specific cardiomyocyte, and the beating rate is very slow, approximately 10 to 30 per minute. So the transplanted ventricular-specific cardiomyocyte does not frequently cause ventricular arrhythmia, in my opinion.
Okay, thank you very much. A question from Ramesh Kumar. Hi, Fukuda-san. When is your target for commercialization in Japan? Is there any target date?
Clinical trial will begin from this September, and probably 2 years is needed to complete the clinical trial. And we hope after 3 years, these can be into commercial. But the government decide, so I cannot say.
Okay, well, there are more questions coming in. Seems like it is a topic that a lot of people are very interested in. It's a question from Vidya. How do you plan to scale up the spheroids, I guess, for transplantation to the human heart? I think you already shared about the robotic system that your team has established.
Mm-hmm. The important thing is that regenerated cardiomyocyte is very small, because the size of the iPS cell is likely to that of white blood cells. But cardiomyocyte adult cardiomyocyte is a huge cells, and after transplanted cardiomyocyte, they show the physiological hypertrophy after transplantation. The size of the cardiomyocyte will be bigger by at least 50 times in compare with the transplanted time. And we will transplant 50 million cells for the first clinical trial. But at the later stage, we will increase the number of cardiomyocytes.
Great, thank you. There's one more question that I see in the chat box from Prem. The question is related to the robotic system that you shared earlier. So your robotic system is handling multiple hyperstacks. Are they considered a closed process? And do you have plans to move to large-scale bioreactors in future instead?
Yeah, this is most important. That machine is first generation automated generating cardiomyocyte. This machine is for 8 patients for 1 month.
Wow.
But we will increase this size to 4,000 patients per year, the second generation machine. We are now preparing.
Okay, great. That's very exciting. Okay, I think I've addressed all the questions in the Q&A box, but perhaps there might be more coming in. But I think that I have a question to ask you, and that relates to what you shared in the later part of your presentation, which is your deal with Novo Nordisk, which I find extremely exciting, and it's very recent. Could you share a little bit more about how this deal with them came about, and in particular, what aspect about your company or the technology drew them? And also, while you were negotiating this deal with them, what were the priorities for you as a startup company?
Hatseed is a startup company, but our technology is very excellent. And during the deal process, Novo Nordisk gave us more than 2,000 questions. It's a tough time, but we had answered every question. And they completely agree with our opinion or with our data. That's why this big deal came out.
Right. I mean, in terms of the priorities for a startup company, I note that for the deal, you will be fully in charge of the commercialization plans and outcomes in Japan, but they do have a exclusive licensing agreement for the rest of the world. Is that right?
Yeah, this is a first step for our company. We have a lot of seeds in our hand, so we will produce more products afterward.
Okay, great. More to look forward to. Another question that I have for you, which is, might strike a chord with a lot of people in the audience. You started off as an academic professor and has been in the academic field for a long time with, deep expertise in the technology. How has the transition to being CEO of a company and a growing company been? So what were the, key challenges that you had to overcome?
About 10 years ago, academic professor, it was very difficult to become establish their own company or become a CEO. But Japanese government changed their strategy about 10 years ago. And they encouraged us to become a founder or a CEO of this startup company. And I'm the success case.
That's great to hear. Hopefully we see more to come, and definitely in Singapore as well. I have time for one last quick question. Hopefully there's a quick answer to it. From SK Chan, is there any risk to the patients, for example, blood clot?
For the first in human clinical trial, we'll transplant the cardiomyocyte from outside of the heart. So in this case, there are no problem. But in the future, we will apply other type of transplantation, such as using a catheter. But in this case, we need to make a more good apparatus or device to transplant the cardiomyocyte. In such case, there is a risk of thrombosis or clot or like that.
Okay.
But we need to overcome these things.
Thanks, Prof. Fukuda. We're not going to ask you 2,000 questions. So thank you very much for addressing all the questions that the audience and myself have. And we wish you all the best with the upcoming clinical trials and the rest of your products in the pipeline as well. So thanks, Prof. Fukuda. Now I'd like to move on straight to Prof. William Huang, and I will hand it over to Prof. Huang for your presentation now.
Thank you so much, Natasha. I hope you can hear me well. And thank you so much, Professor Fukuda. Really, it's my honor to be able to speak on the same platform together with you. And it's really a pleasure to be able to give this talk on this subject for a passion of mine in terms of hematopoietic stem cell expansion. Hematopoietic stem cell transplants have been my profession and my specialty for the past 20, 25 years right now. And this is an area that is increasing in importance. By 2040, we think there will be 1.85 million cases of blood cancers around the world. And these blood cancers are really cancers of blood stem cells originating from the bone marrow. These cells give rise to myeloid progenitor cells, lymphoid progenitor cells, and then giving rise to mature blood cells like white blood cells, which help you fight infection, red blood cells, which carry oxygen around the body, and platelets, which help prevent bleeding. None of these functions we can do without, and therefore hematopoiesis is absolutely essential for life. And through the years, blood cancers have been growing in number. On the right, you will see a chart of all cancers around Singapore, around the world, as well as the increase in leukemia, prostate cancer, and breast cancer. Well, there's been some moderate increase in breast cancer being seen around the world over the years. The increase in blood cancers, particularly leukemia, has been even more stark. The reasons why are not so clear, but certainly leukemia is one of those disorders that are not affected by lifestyle. So even with lifestyle improvements, you can't run away from leukemia. You can't run away from blood cancers despite an improvement in exercise, diet, and so on. So this is an area of increasing concern. Now, hematopoietic stem cell transplants are a way to cure blood cancers by replacing blood stem cells. Basically, we collect donor cells from a donor, and it can be taken from the blood, bone marrow, or cord blood, and taken and processed. And then the patient undergoes conditioning, high-dose chemotherapy without radiation, and the cells are given to the patient. To graphically present it another way, if you imagine this is the patient getting sick somewhat with chemotherapy, and sometimes chemotherapy cannot get rid of the disease, and therefore you need to completely clear the bone marrow system in order to restart it from fresh. And having given him high doses of chemo and radiotherapy, he probably cannot survive because his blood stem cells will not be able to regrow. However, if he receives a fresh infusion of blood stem cells, whether from another person or collected from himself earlier on, that can restore his blood hematopoiesis and re-establish him and cure his blood cancers potentially. But can transplants be better than chemotherapy in curing blood cancers? Well, not all blood cancers need a transplant. But in some of the high-risk blood cancers, some patients with high-risk acute myeloid leukemia, transplants do have a benefit. As well as patients with multiple myeloma who have relapsed after rounds of chemotherapy, they can also benefit from a transplant. Now, currently, it's not possible to do a randomized placebo-controlled clinical trial involving transplants. For certain, you can't placebo a transplant. So in the past, what we have done is to look at patients with a donor or without a donor. So for patients for whom we can find a donor, they are assigned to the transplant group. For whom those patients we cannot find a donor, they are considered the non-transplant group. And you can see that's an advantage in patients who had a donor and who are able to be transplanted. Now, currently, you can see these studies were done about over a decade ago, because currently it's much easier to find a donor for transplantation because we have multiple sources of donors. We can look for cord blood as a source. We can look for half-matched donors as a source as well. And certainly to do a donor, non-donor comparison is very hard to do nowadays. But certainly this is an example of how transplants have been able to benefit patients. Transplants can also benefit patients with autoimmune disease. These are autologous transplants using the person's own blood stem cells. And you can see transplants are superior to disease-modifying drugs in time to disease progression, for example, in multiple sclerosis, as well as time to first relapse as well in this particular instance. Transplants can also be used as a form of enzyme replacement for patients with particular enzyme deficiencies related to blood stem cells. And for some of these disorders like Hurler syndrome, Hunter syndrome, adrenoleukodystrophy, you can see that these patients would otherwise have a dismal survival, and most of them would pass away within a few years of birth. These patients can be cured with a hematopoietic stem cell transplant, and more so if the transplant is done early, the diagnosis is made early, and the transplant is done early before multiple complications set in. So actually, transplants can also cure some of these metabolic hematopoietic cell disorders. So currently, we've had more than 60 years after the first bone marrow transplants. The first bone marrow transplants being done around 1958, prompted in large part because of concerns about radiation catastrophes after the first atomic bomb in 1945. So because of that, a lot of research was carried out to see if the bone marrow system can be restored with a fresh infusion of stem cells from someone else. The subsequent years, there was a lot of experimentation going on before the really successful transplants started to take place with the advent of immunosuppressive therapy, with the advent of HLA typing and so on. But currently, there are over 600 transplant centers in 60 countries and more than 60,000 transplants being performed every year. And currently, over 1.5 million transplants have been performed around the world. And Singapore is a regional referral center for hematopoietic stem cell transplants. And we do well, at least before COVID, we had been getting quite a number of patients from the region. So for sources of blood stem cells, we can obtain them from bone marrow, doing bone marrow harvest involving multiple bone marrow punctures from a donor, collecting usually about a liter of bone marrow from a donor and then giving it to the patient. Or peripheral blood stem cells, where the stem cells can be collected by pheresis, spun down in machine, and then kept for the patient in autologous transplant or given to another patient. Or they can also be obtained from the umbilical cord blood. After the baby is born, the umbilical cord is clamped and cut. So the baby is completely not affected because the process in the clamping and cutting is not modified. And then the baby is put aside. And then whatever residual blood in the umbilical cord before the placenta is delivered is collected as umbilical cord blood for transplantation. So there's been several advantages for umbilical cord blood grafts, resulting in some increase in cord blood utilization in the previous decade. And this is because of a greater tolerance for mismatches. With cord blood, mismatch transplants are possible without any modification of conditioning, without selection of cells or anything more to be done to the cells. It's much easier to find a match for transplants. And the cells are, not only that, the cells are also immediately available. Mismatched transplants have the similar outcomes as fully matched bone marrow or peripheral blood stem cell transplants, and we did a meta-analysis of this before, and also with reduced graft versus host disease. I said before, it's readily available for immediate transplant, and this is an issue we found, for example, during COVID. During COVID, it's sometimes not so easy to get the donor to be available for donation because of various reasons, because of fear of coming to donation centers, because of various reasons. But however, the cord blood unit has already been screened for viruses, has already been typed, and everything has already been done for the cord blood unit. And the cord blood unit in the liquid nitrogen tank cannot change its mind. It's available immediately for transplantation. So unlike some adult donors who sometimes change their mind at the point of donation. So cord blood transplants have been shown to, in this survival done by the Fred Hutchinson Cancer Research Center, in a large study that they did, they compared cord blood transplants against HLA match unrelated donors, as well as HLA mismatch donors. And they found that cord blood transplants perform just as well as fully matched bone marrow donors. Not only that, cord blood transplants in that study was also shown to have lower relapse rates compared to fully matched bone marrow. Now, this was a surprise because people thought that cord blood cells, since they have less graft versus host disease, less prone to attack the patient, may also be less prone to attack the leukemia. But that's not true, because actually, and this is increasingly shown in larger and larger studies, the cord blood transplants actually have lower relapse rates compared to other cell sources for transplantation. This may be because of the naivety of the cells and the ability of the cells to react against bad antigen expression in the leukemia cells. Now, this comparison becomes even more stark if the patient go into transplant with minimal residual disease at the point of transplant. That means they have not completely cleared their leukemia prior to transplantation. And in this case, it exerts an even stronger anti-tumor effect, and relapse rate is even lower in the cord blood group. Consequently, in this particular group of patients with minimal residual disease, survival for cord blood transplantation is better than patients undergoing fully matched marrow or mismatched marrow. However, despite that, cord blood grafts do have a disadvantage. Cord blood grafts engraft slower than peripheral blood stem cell or bone marrow grafts, with an engraftment around 25 days after transplantation. This additional 10 days or so of very low blood counts results in a higher rate of infections and a higher rate of early mortality in patients undergoing cord blood transplants. But bearing in mind that despite this higher early mortality, this is offset by the lower graft versus host disease and the lower relapse rates with cord blood transplantation, resulting in the even and slightly better survival of cord blood subsequent to that. But what if we could overcome this barrier and make cord blood grafts even better and become really the choice graft for transplantation? So one of the disadvantages of cord blood is the cell dose. Cell dose is very important for cord blood transplantation because with a low cell dose, mortality is high. With a high cell dose, mortality is low. And the reason why a low cell dose causes a high mortality is because there's fewer cells, so it takes longer for blood counts to recover, longer for the immune system to reconstitute, and higher incidence of graft failure. So what if we could overcome this through blood stem cell expansion and grow blood stem cells to many more blood stem cells from the cord blood before we infuse them for transplantation? Will we not overcome this barrier and improve cord blood grafts such that they become superior, retain all their advantages, and become superior to the other modalities? However, this is easier said than done because growing mature cells and growing increased cell numbers is so easy to do. We can do it so easily in the laboratory. But in order to grow true blood stem cells is really tough because cells will tend to mature and they tend to grow into just more mature cells. So getting the true blood stem cells phenotypically and functionally defined is difficult to do. However, this is increasingly shown to be a possibility. This is a cord blood expansion study that we took part in. This is by a company called Gamida Cell, and we took part in the NICOT phase 2 multi-center trial as well as their phase 3 study. The cord blood unit is separated into two fractions. One fraction is expanded and the other fraction is the non-expanded cells that is subsequently infused together. And in this particular study, it was shown that engraftment is much faster compared to the usual 21 to 25 days after transplant for neutrophil engraftment. It was accelerated to 11 days after transplantation, even better than peripheral blood or bone marrow transplants. And consequently, survival days alive and out of hospital in the first 100 days post-transplant is improved to 73.7 days compared to 52.9 days. This is, in other words, the patient stays for about less than a month after a transplant compared to 2 months in hospital after a transplant. The phase 3 study, which is just published, shows a similar pattern, but with also indications of better overall survival in the patients undergoing the cord blood expansion group. Okay, can we make this even better? And I'll outline some reasons why we want to make it better. Now, this is, I was very fortunate to work with this team, Dr. Christina Chai, Gigi Chu from NUS, Department of Pharmacy, and this is Sudipto, who did most of the work, which I will show you later, and Qising over there also did a lot of the work for the synthesis of the molecule that we will talk about. And this is really to meet the unmet need in stem cell transplantation, in terms of allogenic, autologous transplantation, and gene therapy, which I'll talk a little bit more about later. So there are current limitations of competing technologies right now. A lot of them require prior selection of CD34, hematopoietic stem, and progenitor cells. And all the studies, including the gamete cell one, require this. They also require a high cell dose of CD34 stem cells to obtain significant expansion. And one of the issues in the study that we took part in is that we ended up having to select very high cell dose cord blood units in order to be able to qualify the cord blood units for expansion. In some cases, it became difficult to recruit some patients because we could not find sufficiently high cell dose cord units available for them. Many of these protocols also support the growth of leukemia stem cells, and the safety profiles of some of the small molecules were not so clear. So in collaboration with the group that I showed you in the previous screen, we developed a non-azole small molecule called C7 that expands hematopoietic stem and progenitor cells. We did multiple studies. I showed it to be safe on the study panel that you see on the screen. We showed that the cells could significantly expand in the blue bars, cord blood hematopoietic stem cells, compared to our conventional cytokine controls. And these are for CD34 positive cells. And not only that, they could also expand extremely rare populations of cells, which are CD34 positive, 38 negative, 90 positive, and 49F positive cells, thought to be the true hematopoietic stem cells. And this we had never ever seen before, and we had never ever seen a flow plot shine so bright in this particular area before. I mean, we compared our molecule against some of the other molecules that others have explored for hematopoietic stem cell expansion, and found it to be superior compared to some of these currently in clinical trials. We also found that we were able to engraft NSG mice, not skip gamma mice, with stable engraftment in the mice in the bone marrow, not only in primary transplantation, but also in subsequent secondary mouse transplantation, showing stable engraftment of the cells. How does it do it? This is the ongoing study in terms of the mechanism of action, but certainly C7 increases cell viability of the cells, causing the cells to be more viable compared to if they were not in this C7, and also decreases mitochondrial superoxides, again, indicator of cell health and viability. And there are various studies that we currently have ongoing in terms of 10x genomics and multi-parameter flow that we will be able to elaborate on in the near future. So our current blood stem cell technology, which I don't have all this time to be able to show you, does not require prior selection of CD34 hematopoietic stem cells. We actually do not require a high dose to start off with. We can actually start with pretty low doses to obtain significant expansion. We also show that it does not support the growth of leukemia stem cells, which I'll show in another slide later. The safety profile is clear, and we can also do expansion from adult sources as well. So in the near future, we are looking at processes for developing next generation grafts using laminins and non-azole small molecules. So this is our data on not expanding leukemic stem cells. And if you just the black bars are basically DMSO control, but the slightly orange bars, you can see with increasing doses of C7, the leukemia cells actually die on the higher doses of C7, showing that not only does C7 not promote the expansion of leukemic stem cells, but they're actually detrimental to leukemic stem cells, but beneficial to healthy hematopoietic stem cells. And this is important because it's relevant not only to the fact that it does not cause leukemogenesis, but it is also relevant because if we were to extend this into autologous transplantation, it becomes even more attractive because if we were to grow stem cells from a patient needing his own blood stem cells later, at least we are confident that we will not end up growing his blood cancer cells at the same time. So we currently have this proprietary azole small molecule. It's a proven stem cell phenotype, accelerated and sustained engraftment in animals with mechanism of action studies ongoing, and so safety and toxicity studies which have been done. And compared to competitors seeming to have a higher expansion of the CD34 bright population compared to other competitive technologies. And we think that our graft will be able to help improve short-term issues of infection risk, associated mortality and hospitalization costs, while also reducing graft failure, reducing complication, reducing relapse, and high healthcare costs associated with the long hospital stay while awaiting engraftment. So this molecule is currently being produced in large scale as a raw material for GMP manufacturing, and currently process development is ongoing for the expansion into GMP for our upcoming clinical trial. So about this clinical trial, I'm really delighted to say that I was very encouraged with this collaboration. This is a first-in-man clinical trial that's been supported by the National Medical Research Council of Singapore. And it took the collaborative effort from Duke NUS. This is Chris Lang, who leads the innovation and entrepreneurship at Duke NUS, and also CTED, the Center for Technology and Development. It's a landmark clinical trial. Also collaboration of our clinical PI, Dr. Francesca Lim from the Singapore General Hospital. And most importantly, a private-public partnership with CordLife to bring different players into the ecosystem to enable early phase clinical trial to de-risk technology, and so that we can obtain safety, efficacy, and manufacturing feasibility data in our first pilot study. Now, if this turns out to be effective, blood expansion in our study and others turns out to be effective, the implications are myriad. This is quite a realistic bell-shaped curve of the cord blood units that are donated, and some of them banked in cord blood banks around the world. Now, most of the units are in this kind of cell dose range. Some of the units are discarded below this cell dose, or even below this cell dose, because nobody will use them. Because majority of the utilization of the cord blood units will be in a high cell dose range, because low cell dose units are not wanted by transplant doctors. And being a transplant doctor myself, I wouldn't choose the units anywhere here. But this is where the majority of banked cord blood units are. And it's a wasted resource in many cord blood banks around the world. And we can't help this as transplant doctors. We do require that, because below the cell dose, like I said, mortality for the patient is high, and we wouldn't want our patient with the high mortality. So you can just do a two-fold expansion. You can push a cord blood unit from the low utilization to the high utilization zone. And in fact, our technology shows that we can do multiple fold expansion of this. So certainly the advantage of this is going to be tremendous in terms of the ability to use cord blood units in cord blood banks around the world that are otherwise would have been languishing. In addition to that, we think that it's also applicable for patients undergoing an autologous transplant, for example, if we have insufficient cells after a bone marrow or peripheral blood stem cell harvest. And this is particularly where the C7 is particularly important, because the molecule does not promote the expansion of blood cancer cells. So, and therefore in autologous transplantation, you can you have assurance that that will not happen, and you will just expand the true blood stem cells instead. Furthermore, sometimes in transplant from another individual, sometimes high numbers of cells are needed for cell selection procedures, and sometimes not enough can be collected, especially when the donor is much smaller than the patient. And in that case, to get sufficient numbers of blood cells in an allogenic transplant, or even to make the allogenic transplant safer, blood stem cell expansion may be an important component. Maybe in the future, we won't need to be asking donors to donate a liter of their bone marrow each time, but maybe just a much smaller amount than that may be sufficient for patients. This improves safety not only for patients, but also for donors. Can also be used as a platform for gene therapy because blood stem cells can be used to treat patients with thalassemia, sickle cell disease, and many other disorders. But the problem in allogenic transplantation is that you're putting someone else's immune system into the patient, and that could cause side effects of graft-versus-host disease as well as other disorders, even if it's from cord blood. But however, if you can take the patient's own cells, correct the gene and give it back to the patient, this is an area that's been increasingly explored for various disorders to treat various genetic disorders. But the problem in this is that it's going to be slower because when you do the gene correction, only a few cells will have the gene corrected. And in order to exert the beneficial effects, it will take much longer for these cells to grow into the required amounts to correct the genetic disorder. So if blood stem cell expansion could be added to this after the gene correction, then certainly, or during the gene correction, then certainly we would have a much more efficient gene correction procedure as well as a much more efficient gene correction transplant. In addition, even if we don't expand blood stem cells, you could just increase the number of cells given to patients, even they don't last for a very long period. It can be used as off-the-shelf therapy to reduce the period of neutropenia after patients undergoing intensive chemotherapy. And I would say this is particularly beneficial for patients with some of the blood cancers like acute leukemia, where chemotherapy is particularly intensive and where I've seen many patients suffer from bacterial infections, fungal infections, and other infections because of the neutropenic period that we take it that it's a given that it has to happen. But maybe this does not have to happen at all. And maybe transplants in future can be done much more safely as outpatient with many of these risks reduced. So with that, in this very short presentation, I'd like to thank the various people who have helped us along this way. There are others, I'm sure, that I may not have added into this thank you, but certainly very grateful for the different collaborators around the island, including collaborators from NUS, the Singapore MIT Alliance, the ACTRIS, the Advanced Cell Therapy Research Institute of Singapore. And really happy now to take any questions on this presentation. Thank you.
Great. Thank you very much, Prof. Huang, for your presentation. And I think I was very intrigued by all the data that you presented. And like you mentioned, the applications for your HSC expansion technology is tremendous. So we have a few minutes for questions. There is one in the Q&A box by Vin. So the question is, what is the mechanism why C7 only works on normal HSCs but not leukemia cells?
I would say, Vin, that is a very good question, a very important question. I don't have the answer to it, but interestingly, C7 is related to some MAP kinase inhibitors, although I don't think it works by MAP kinase inhibition because we tried other MAP kinase inhibitors and it does not. MAP kinase inhibitors do not work in expanding blood stem cells. So I think there's some anti-leukemic effects among the C7 effects, including P53 as well. But interestingly, while it prunes off the less ideal cells, the cells that are detrimental to cell culture, and including leukemic cells, it leaves the true hematopoietic stem cells untouched. I would say it's like the growing of a vineyard. When you grow a vineyard, you want to prune off the leaves and the vines that aren't doing so well, so that the healthy ones will grow better. But that's a bit of hand waving, because I would say that we don't have the mechanism fully worked out yet at present. We're going in depth right now and doing multiple pathways that we're looking into.
Okay, great. Thanks, Prof. Huang. I don't see any others in the Q&A box, but I have a couple of questions. I mean, maybe I'll just ask the first one. I mean, you sit on numerous, national/public programs, and you work across multiple collaborations. Can you comment on how these collaborations, and also how your public-private partnerships, you mentioned CordLife, how do these accelerate the development of the technologies through to clinical translation? And how do you think scientists and people in Singapore can establish more of such partnerships in an effective manner?
Yeah, I would say that science is really very much a team sport, right? We are good in what we do, but we can't be good in everything. And I must say I'm really very grateful for the way in which the ecosystem here has grown in many ways, such that there are many players in the ecosystem who are willing and ready to help, and willing and ready to be a team partner in this journey. And like, for example, we had, I think the most important part in this entire partnership is, of course, to engage clinicians. I mean, I myself am a clinician, but I have to make sure I'm not so biased, right? And engage my colleagues to see if they buy into this or not. To ensure that they see there's a need, there's a gap, to find out what they want to see in terms of the cell product, in order to really want to be able to use it, and to also to be able to identify a driver who will be able to drive this clinically independent of my cell. On the other hand, there are also many technology facilitating bodies, whether in A*STAR or otherwise. And like in Duke NUS, we have CTED, Center for Technology and Development, which is able to help us craft a financial case. We also had Candy, who's with us in NCC, who's was able to help us do that, to also help us craft our pitch. Basically, as a clinician, or sometimes as scientists, when we talk with many caveats, right? We say yes, but this, and we don't speak so fluently or efficiently. Basically, to craft that pitch that's simple, direct, and something people can relate to. But in truth, everyone I found that I've encountered has, including the venture capitalists, have a desire to make an impact. Everyone, besides, it's not just about wanting to make money and things like that. People, their reason for existence is to make an impact. And a lot of the impact has to do with, making a difference to the lives of people, and making the difference to the lives of patients. So I think in that regard, we're quite grateful for that. And I would say that we're quite grateful to see collaboration from CordLife, for example, come on board to support this clinical trial with the corresponding matching funding from the NMRC. It's also an example of private-public partnership that I'm really welcoming in this regard. And in this regard, I think that this ecosystem that's been created has allowed the rollout of some companies linked to us, like Tesla Therapeutics, Lion TCR, and otherwise in the past. But of course, at the end of the day, after this technology rolls around, as a clinician, I also find that then there's the issue of costs, right? How much will this cost for patients, right? I mean, everyone has an idea of what it needs to be, and of course, the companies need to recoup their costs, right? So then it involves engaging governments, engaging insurers, engaging clinicians, and gathering enough evidence to say that it's worth it, for the patient. is worth it compared to other alternatives that you might put the patient on. And actually, it's maybe even cost savings. And in the regard, we are very actively engaged in writing of some cost effectiveness papers as well. And I would say that at the same time, in this entire ecosystem and in all this teamwork, a lot of projects may not succeed. And that's the truth. But it's important to recognize that it's not all about the destination, it's also about the journey. And what we learn from each journey. I've also had a previous study before where I dropped, for cord expansion because the results were less than ideal. But it's important to recognize that early and to drop it. But what you learn from the journey, a previous less than successful journey, puts you in good position for the next expedition.
Okay, great. Well, we still have quite a lot of people on the line, so thanks for staying throughout. And I guess we'll just have one last question. I mean, you answered quite a lot of the ideas that I had in my head just now with your last answer. But from your perspective as a clinician treating patients, I know you talked mostly about blood cancers and of course your blood stem cell technology. But for all the rest who are working on other types of therapies or other technologies that address other diseases, what do you think scientists or clinician scientists should take note of to enhance the success of translation? I mean, is it a lot about the preclinical success or the manufacturing capabilities or the wide range of applications that the technology can have? Like what do you think are important aspects that scientists can consider?
Well, that's a very involved question.
Okay,
because
yeah.
although it sounds simple, it's actually very deep, because the thing is, it depends on how you define success, right? And for some, success is in getting very high impact papers published, and for some, success is in getting a company spun out with millions of dollars. To some, a success is in making sure that the next patient is very well treated, and that the patients benefit. So all of us define success somewhat differently, and rightly so, because there should be many different types in the ecosystem. I would say one area sometimes that is difficult is that we may not have currently refined this translational journey in such a way that it's like a process manufacturing, in a sense that right now, if a researcher develops something, finds something in the lab, he is bringing it to the next phase, he patents it and so on, and then he publishes. And then sometimes it dies a death there because he or she may be less sure what's the next step ahead, right? Whereas the next step ahead may be someone else should take that next step. And then subsequently, when it progresses beyond that next step to involve a VC or otherwise, there may be subsequently the manufacturing, someone else should take on that next phase. And then subsequently, when it needs to go to the clinic, again, it's maybe the clinician, a different kind of clinician, a clinician driver, who should be taking on that next phase. Now, currently, the system seems to expect the same person to take it from beginning to end, and somewhat challenging because for every person trying to do it, he's doing it for the first time. And as we refine this process of translation a little bit better, maybe this can be more of a handover with the continued stake in the entire thing to someone who's more experienced in doing the next phase. But of course, I mean, no one loves your baby more than you do, because if it's your baby, you'll make sure it grows.
Yeah.
Yeah.
Okay. Well, great. I mean, that's a great answer. So thank you very much. I mean, I hopefully, through your various involvements, you'll be able to mentor and advise some startups or some budding clinicians or scientists that are going to take this journey. With that, I think we are done with all the questions and the time is up. So thanks again to everyone who stayed. Thanks to Prof. Huang and Prof. Fukuda for your presentations and for taking the questions. And I will hand over the time back to Jody to close the session.
Thank you, everyone. Thank you, Prof. William Huang, Dr. Natasha Ng, and Professor Keiichi Fukuda for staying with us, as well as for the 70 over attendees that we still have on the line. I'll keep my closing short, but thank you again to the presenters for sharing such informative discussions on your innovations. And for our attendees as well, please keep a lookout for our post-event EDM, which will include a video recording of our session for you to share with your friends and network, as well as the presentation slides by both professors today. I think that everyone actually enjoyed today's session, so please keep a lookout for more sessions like this and join our events at SGInnovate. And lastly, I wish everyone a great evening or afternoon or morning from wherever you're tuning in from. Thank you, everyone, and goodbye.
Okay, thank you.
Thank you. Bye-bye.
Bye.
Bye.